Good evening. We'll be starting in a couple of minutes. Let everybody join in. We'll wait for 1 more minute and then we'll start. Let's see if anybody is there to join. Welcome to the week 6 of the interactive doubt clearing sessions for the course Genome Editing and Engineering uh, in NPTEL. This course is taken by Professor Utpal Bora of IIT Guwahati. And I'm Sanket Mukherjee, Prime Minister's Research Fellow from the School of Biological Sciences at IIT Delhi. So let's talk about the summary of the week 6 about what we have discussed. So we'll talk about TAIL, that is transcription activator like effector. These are very nice DNA binding domains. We'll talk about it. We'll then discuss about a little bit about their discovery and the delivery methods. And then we'll talk about designing, you know, talents. So these are transcription activator like effector nucleases for genome editing. So going forward, about a brief history of talents. So as you can see, the proteins in the blue, they represent talents. Sorry, tail in specific transcription activator like effector molecules. So these are repeats which have different um, RVDs. These are dinucleotide, divariable units which give these proteins specific information of which DNA to bind to. So these they show different DNA base pair preferences, and consecutive RVDs in a tail correspond directly to a DNA sequence in the binding site, resulting in a simple one repeat to one base pair code. This is because of it is because of this reason why tails could be engineered um, to recognize DNA of our own interest. So that's why it's very important for genome editing. So the basic building block of tail protein is a helical hairpin structure. So these helical hairpins, it has around 34 amino acid, 34 repeats. We'll talk about it in detail. So the helical hairpin is the basic building block, and tail protein, they contain a series of alpha helical hairpin domains. We'll talk about it. So it was first, discovered together in by two groups. One was Matthew J. Moscow and Adam J. Uh, Bornanov, and then Bosch and group. They discovered the role of tail in causing infection in plants by a pathogenic bacteria called Xanthomonas. We'll talk about it. So uh, how does it work? So in case of a Xanthomonas species, what we observe is that they have these effector proteins which are secreted through type 3 secretion system. So we'll talk in details about these different types of secretion system which is present in a bacteria. So these effector proteins, they are secreted via the type 3 secretion system, and they are directly delivered to the plant cell cytoplasm. Now, these effectors, they consist of transcription activator like domains which induce transcription. These are DNA binding proteins that upregulate the plant genes, which lead to further susceptibility. So in case of zoo-rice interaction, the classical example is the upregulation of sweet gene. So another class of T3S effectors are non-TAL effectors. They are involved in dampening the immune response by targeting the defense signaling pathways. working directly or indirectly by binding to plant proteins. So the plants, they also confer resistance to these kind of effectors. Uh, so uh, because of which the plants, they also develop a resistant mechanism against these effector molecules. So this is how it works out. And if we talk about some bacterial secretion systems, so there are six, in this case, seven actually has been shown, but six essential bacterial secretion systems, which either are have a secretory function or a translocation function. These two are important. So the pathways of T1 secretion system, T3, T4, and T6, they contain periplasm spanning channels and secrete protein from the cytoplasm outside the cell. However, the mechanisms of each of these secretion system is different. Three of these, like T3 secretion system, T4 secretion system, and T6 secretion system, can also transport protein across an additional host membrane. In this case, it would be a plant cell. It will deliver the proteins directly to the cytoplasm of this target cell of the host. Now, this is just a brief overview about the different secretion systems that are present in case of Xanthomonas. So, like type 1 secretion system is important for the adhesion of the bacteria to the host cell or the extracellular matrix. Type 2 secretion system is important for host cell wall degradation. Type 3 secretion system is important for host cell manipulation and phytopathogenesis. So this is what we are more concerned of, because this is the type of secretion system involved in case of transcription effector like effector molecules. So then transcription T4, type 4 transcription, type 4 secretion system, they are important for combating bacterial competition and conjugation. Type 5 secretion system, it is important for adhesion to host cell or extracellular matrix and auto-regulation, like auto-aggregation, like formation of biofilms and structures like that. And type 6 secretion system is important to prevent any microbial competition in case in infecting the host. So Xanthomonas, it harbors almost all types of secretion systems, right from type 1 to type 6. And this is where we discovered talents initially. So regarding the T3S system, so it spans in both the bacterial membrane and is extended by a hollow conduit, which is the HRP pilus. So this HRP pilus is the it's kind of an extension of the bacteria, which allows this effector molecules or other kind of phytopathogens, pathogenic molecules to get into this plant cell wall. Now, bacterial effector proteins are secreted there through the T3S system and translocated into the plant cell cytoplasm via bacterial translocon complex that inserts, let's say, in this case, it could insert proteins into the plant plasma membrane. So this is how it works out. And once this proteins, these effector molecules, they are inside the plant cell, they can perform various functions. They could upregulate gene expression. They could cause hormone signaling. They could cause degradation of certain proteins. So there are many other things which it can do. So to talk briefly about the structure of tails. So again, tail is a transcription activator like effector molecules. They essentially have 1834 amino acid repeat modules. So these 34 amino acid repeat modules, they only form the helical hairpin structure. And within these repeats, the 12th amino acid and the 13th amino acid, which are marked in red in this diagram, they decide the specificity of binding. So to which particular base will this DNA binding domain bind to, that is decided by the 12th and 13th residue among this 34 amino acid repeats. So in the C terminal, if this is the N terminal and this is the C terminal, if we see it like this, then the C terminal, it contains three nuclear localization signals. So this is important because this, if the nuclear localization signal will determine the fate of this protein to get into the nucleus. There are sometimes mitochondrial localization signals also. So they are called MTS. So mitochondrial localization signal will cause the protein to get into the mitochondria. So this is how the localization signals work. So because of this nucleus or nuclear localization signal, the proteins are imported to the nucleus. Now, downstream of the nucleus localization signal, there is an acidic activation domain which works as a transcriptional activator. That's why the name comes as a transcription activator like effector molecules. Now, there are 34 amino acid repeats which bind to one base pair of DNA. The specificity of these repeat, I mean, of this binding is, determined by the 12th and 13th amino acid position, which are hypervariable residues. They vary a lot, and these are called repeat variable die residue, RVDs. Now, these repeat types have specificity for more than one or for one or more than one base pairs. So so during this process of transcription activator like effector molecules binding to DNA, there could be two mode of actions. One would be the search state, and the second would be the recognition state. So in the search state, what happens is um in this process, in this time, these molecules, they search for binding and they traverse across the DNA as shown in the above figure. And there could be some ineffective binding that would cause steric hindrance, and that's why they are extended. They have an extended structure. It's kind of a relaxed form. But once it finds or it recognizes the place where it needs to bind to, it then compresses itself, and the superhelix is now compressed, and it now binds to the specific DNA of interest. So that's how it works. Now, it is because of this property of transcription activator like effector molecules that they can be designed to bind to any kind of DNA sequence, because previously we saw that there was FOC1. So FOC1 did not have uh, we cannot engineer them to have multiple kinds of DNA binding sites. But in case of talons, uh, we can just change the 12th and 13th amino acid residue, and we can basically uh, design a particular transcription activator like effector molecule which will bind to our DNA sequence of interest. So it is because of this property, these tails, they were used along with, they were fused with, let's say, repressors or activators or nucleases, methylases, or other kind of molecules. So they give um, a lot of control. How do you want to use these molecules? So if it is bound to repressors, then you could cause gene knockdown. It's used in case of crab, let's say. And then you can use activators for gene activation. Then you could use nucleases, like that's why it's called talents in this case, which is used for gene modification. Then methylases could be used for epigenetic research if we want to methylate a specific region. If we know the sequence of the region, we can do that, and we can do other things as well. So for delivery of talents, they could be delivered in their DNA, RNA, or protein form using various methods. There could be physical methods like biolistic transformation, microinjection, electroporation. There could be bacterial methods. There could be viral-based delivery methods like adeno viral vectors. Then there are retroviral vectors, adeno-associated viral vectors, things like that. And there could be chemical methods that utilize transfection reagents, liposomes, or polyethylene glycol. These are all, agents that allow the protein to get inside the cell. So the choice of these delivery methods, they are based on what is the aim of your experiment, and what which kind of organism you're trying to target. So based on that. Um, so again, this slide is about the architecture of talent. So again, usually what we do is the DNA binding domain of the transcription activator like effector molecules are used for recognition of the DNA, and then it is fused with another restriction enzyme. It could be FOC1 or PVU2. PVU comes from Proteus vulgaris. So based on that, if it binds to FOC1, then there is, as we know that FOC1 cleaves the DNA, it will cause a double-stranded break. Similarly, PVU2, it will again cause a double-stranded break to happen. These tails could be, fused with meganucleases, like truncated version of meganuclease TAVI, as in this case. Um, it is encoded from bacteriophage T4. So we can use meganucleases for this. Then there could be a full length sequence of meganuclease. So truncated version or full length version, we can use meganucleases to cleave the DNA and cause a double stranded break. NICs or a single stranded break could also be introduced using tails. So here we used different functions, different proteins like MUTH or again TEV1, the another truncated version of meganuclease. And MUTH is a DNA mismatch repair proteins. We can fuse them with transcription activator like effector molecules, and we can create this kind of mutants, which could cleave or double-stranded DNA. Which could cause double-stranded break. So that's the idea here. That's how we can engineer transcription activator like effector molecules. Beyond this, there were multiple computational tools, multiple methods of assembly of transcription activator like effector molecules, so that we can get our own let's say we can create a recognition domain which will bind to the DNA of our interest. So to do that, there were multiple, technologies that came up after 2010, because that's when tail was discovered. So the question will come to certain questions. So transcription activator like effectors are produced by a virus, be archaea, C bacteria, or D fungi. So I think we'll give 2 minutes for people to think and they can answer. So the answer to this would be a bacteria, especially Xanthomonas species, which causes transcription activity like effectors to be produced. And that's how it was discovered. Coming to the next part, tail proteins, they contain a series of dash domains. Are they the repeat variable diresidues, TAL effector molecule DNA reading domains, or alpha helical hairpin? So the answer would be alpha helical hairpins. These are the repeats, the 34 amino acid repeats which are present in case of tail molecule. And among these repeats, the 12th and 13th residues, they are called the repeat variable diresidues, RVDs, that are important only for the specificity of this DNA binding domain to bind to a specific, a specific sequence. But these repeats are actually alpha helical hairpin repeats. So coming to the next question about the answer. So talents, they consist of a series of alpha helical domains. So as you can see in the figure in the right, see each alpha helical hairpin domain of tail protein binds to an individual DNA base. And overall, a single tail protein recognizes a very specific long DNA sequence if we can engineer them like that. So tails are virulence factors of plant pathogens like Xanthomonas that are translocated via a type dash inside the plant cell, type 3 secretion system, type 1, type 2, or type 4. We'll give a minute for uh, people to think about the answer. So the answer would be type 3 secretion system, which is how tails work. So we have talked about it. So regarding the different secretion system, like type 1, they are used for adhesion to the host. Um, Extracellular matrix. Type 2 is for host cell wall degradation. Then we talked about type 3, which is important for phytopathogenesis. We also discussed about VHRP pilus, how it is important for transferring the proteins from the bacterial cell to the cytoplasm of the plant. That's how it works. And for type 4, secretion system is important for bacterial competition and conjugation. while type 5 is again important for biofilm formation or autoaggregation of the bacteria, or for adhesion of the bacteria to the host cell. Again, very similar to type 1. It also helps adhering the bacteria to the host cell or its extracellular matrix. And then there are type 6, expression secretion systems, which are important for these for the bacteria to kill any competition that might also be there. So that's important for their survival. That's how they evolve. So talking about the next question, which of the following is not a delivery method of talent into living cells? There are four options: physical methods like biolistic transformation, microinjection, or electroporation; bacterial-based delivery; plant-based delivery; or chemical methods that utilize transfection reagents, liposomes, or PEG, polyethylene, polyethylene glycol. So we'll give a minute for people to think and answer. Okay, so the answer to this question would be plant-based delivery. Plant-based delivery is not used for delivering talents into the cells. Rather, physical methods like biolistic transformation, microinjections, electroporation is used. Bacterial-based delivery is used. Chemical methods like utilizing transfection reagents, liposomes, or polyethylene glycol is also used. Apart from this, viral vectors are also used. So viruses or viral vectors are used for delivery as well. So this is essentially what is given as the answer. The talent can be deployed in form of RNA, DNA, or protein using various delivery approaches into the cell. That could be physical methods like biolistic transformation, microinjection, or electroporation. It could be bacterial-based delivery, viral-based delivery. So among viruses, they could be adenoviruses, adeno-associated viruses, or retroviruses, which would be used for delivery approaches. There could be chemical methods that utilize transfection reagents, liposome, or polyethylene glycol for delivering the talents into the cell. Coming to the next question, full form of STAR is sequence tail assembly reaction, simple tail assembly reaction, simple talon assembly reaction, or simple TAL assembly reaction. We'll give a minute for everybody to think on this. So the answer would be uh, simple tail assembly reaction. So regarding this particular feature, a particular technology which was developed in the year, if I'm not wrong, it was discovered in 2016. So STAR, it addresses some of the shortcomings of the existing approaches like Golden Gate or solid phase assembly protocols, and it enables routine production of tail transcription factors for diverse application in mammalian stem cell and synthetic biology. STAR uses an isothermal assembly, also known as Gibson assembly, that is labor and cost effective, accessible, rapid, and scalable. So this is all about STAR. So a small 68 part fragment library is employed, and the specific tail repeat sequence is generated within 8 hours. So it reduces the time a lot. A sequence verified talons or tail TF plasmids targeting 17 base pair target sequences can be produced within 3 days without the need of for stepwise intermediate plasmid production. Gogolok and Associate demonstrated the utility of STAR through production of functional tail transcription factors capable of activating human SOX2 gene expression. So coming to the sequence of the tail assembly methods, which of these is the correct sequence? So is it 2011 detail, then 12 do it yourself, 13 flash, and wait, let me see. Okay. Or is it 2011 ICA, 2012 Detail, 2016 DCTA, 2017 Start, or is it 2012 Flash, 2013 Do It Yourself, 2016 Star, and 2021 DCTA? Or is it 2011 Detail, 2012 Flash, 2016 Star, and 2021 DCTA? I'll give a minute for everybody to solve this. Think there is some issue with this slide. Let me just stop sharing and start again. Yeah. So just to give you the options once again, just a minute. So is it 2011 Detail, 2012 Do It Yourself Protocol, 2013 Flash, and 2016 Star, or 2011 ICA, 2012 Detail, 2016 DCTA, and 2017 Star? Or is it 2012 Flash, 2013 Do It Yourself Protocol, 2016 Star, and 2021 DCTA? Or is it 2011 Detail, 2012 Flash, 2016 Star, and 2021 DCTA? So the answer to this would be the option D. Coming to the way it was discovered, though in 2010, the role of tail was first discovered in the plant Xanthomonas, and then in 2011, detail was discovered, after which Flash and ICA was discovered in 2012. 2013, do-it-yourself protocol was formulated. In 2016, STAR was formulated, and the latest one is DCT in 2021. So coming to the next question, Lauren Tesson and colleagues first successfully created a gene knockout rat using customized style effectors in the year 2010, 11, 12, or 13. So I'll give a minute for you to think on this. So the answer to this would be 2011. So knockout rats were generated by embryo macroinjection of talons. This paper came out in 2011 by the group of Lauren Tesson uh, and colleagues. So coming to the next question, HIV-1 infection of CD4 T cells, it involves binding of the viral glycoproteins GP120 to the primary cellular receptor dash and either of the co-receptors. So the options are CCR5, TEL32, CD4 or CXCR4. CD3, CXCR4 or CXCR5, CD4, CCR4 or CXCR5, or CD4, CCR5, or CXCR4. Just giving a minute for everybody to think. Okay, so the answer would be it binds to the cellular receptor CD4 and the co-receptors are CCR5 or CXCR4. So if you see the diagram here, this is we have discussed in detail about the replication cycle of HIV. So it binds to the CD4 receptor in an immune cell, and there is a co-receptor CCR5 where it binds to, which causes the internalization of this virus. If the there could be another way also. So apart from CD4, it could also bind to another co-receptor, that is CXCR4. So if it binds to this receptor, then this virus is again internalized, like we have already seen previously. Then also it could be internalized. So we have already seen if there are mutations like del32 base pair mutation in case of CCR5, it prevents the virus to enter into the cell. Hence, the people who have these kind of mutations, they are kind of immune to HIV infection. Coming to the next slide, a mitochondrial targeting signal is fused to the dash of talon to allow localization inside mitochondria. Will it be N terminus, C terminus, N terminus or T terminus or C terminus or A terminus? I'll give a minute for everybody to think. Okay, so the answer would be N terminus. So N terminus has, if this is the C terminus, this is the N terminus, as you can see here, given in the diagram. So the N terminus, it contains the mitochondrial localization signal or the nuclear localization signal, which allows these talons to enter to the mitochondria or the nucleus based on these signals. So the full form of crab and tail crab is knockdown associated box, Kruppel associated box, Kruppel assembly base, knockout regulating associated box. So just giving a minute for everybody to answer. Okay, so the answer would be Kruppel associated box. So about this, so transcription activator like effector molecules, they are fused with some other functional domains instead of nuclease to regulate the gene expression. So in this case, it can be fused with repressor domains. So one of these repressor domains are called Kruppel associated box repressor domain, which when are fused by assembling the tail DNA sequences with that of the transcription activator or this repressor. So Zhang et al., they developed a multicolor panel of lentiviral tail crab expression vectors for knockdown of multiple gene targets. It was successful in the targeting two genes, c-KIT and PU.1 in bone marrows of recipient mice. So c-KIT is a tyrosine kinase receptor, and and mainly is expressed in hematopoietic stem cells and multipotential progenitor. PU1, also called SFP1, I1, is an ETS, erythroblast transformation specific family transcription factor, and expressed in hematopoietic stem cells as well as multipotential progenitor cells. So, a multicolor panel of lentiviral tail crab vectors was constructed by Zhang and its group, replacing EGFP with the genes carrying cerulean, M. cherry, Iris blast, Venus, Iris eosin expression cassettes. The resulting vectors express four fluorescent proteins. And in addition, PLV tail crab, M. cherry blast, PLV tail crab, Venus eosin vectors were equipped with two drug-resistant genes, blast and zeosin. So it is important for selection as a selectable marker. So we discussed about all the questions. Just for everybody, we'll go through them again. So question one was tail proteins. They contain a series of alpha helical hairpins. Then we talked about question number two. We didn't discuss question number one, did we? Okay, so question number one was transcription activator-like effectors are produced by bacteria, especially Xanthomonas species in this case. Tail proteins, they contain a series of dash domains. They contain a series of alpha helical hairpin domains. Then we talked about question three, tails. These are virulence factors of plant pathogens like Xanthomonas and are translocated via uh, type 3 secretion system in case of inside a plant cell. So then we talked about which of the following is not a delivery method for delivery of talent to the living cells. Plant-based delivery method is not a method. Then we talked about the full form of STAR. It is simple tail assembly reaction. It was discovered in 2016 for tail assembly. Then we talked about the sequence of tail assembly methods. So in 2011, detail was discovered just after the discovery of tail in Xanthomonas species. Then in 2012, Flash was discovered. In 2016, Star, and in 2021, DCTA was discovered. This is the sequence as is given. So Lauren Tesson and colleagues, they first created a gene knockout track using customized style effectors in 2011. Then this is the paper about it. HIV-1 infection of CD4 T cell involves binding of the viral glycoprotein GP120 to the primary cellular receptor, uh, CD4, and either of the co-receptors, either CCR5 or CXCR4. That would be the answer. A mitochondrial targeting signal is fused to the N terminus of talon and allows localization inside mitochondria. So full form of crab in tail crab is scruple associated box. So that would be all for today's session. We will be ending it here. We will be joining again next week on Tuesday from 6 to 8 p.m. Thank you.